Good afternoon, dear colleagues and guests. Today, the 15th of January 2021, 4 a.m. Moscow time, we begin the Dissertation Council for the defense of Alexandrovna Mikheenko thesis, the candidate of Physical and Mathematical Sciences in the scientific major 010903, Mathematical Biology Bioinformatics, on the topic Development of Computational Methods for the Analysis and Visualizations of Eukaryotic Genomic Assemblies, be considered open. By order of St. Petersburg State University, dated October 28, 2020, number 9609/1, I, Podgorno Olga, Doctor of Biological Sciences, Professor of the Department of Cytology and Histology of St. Petersburg State University, was appointed the chairperson of the Dissertation Council. The members of the Dissertation Council were approved by the same order. Let me introduce them. Tatiana Valerievna Matveyeva, Doctor of Biological Sciences, Professor of the Department of Genetics and Biology, St. Petersburg State University. In the remote access mode, we have Gelfand Mikhail Sergeyevich, Doctor of Biological Sciences, Vice President of Biological Research, Skolkov Institute of Science and Technology. Can you hear and see us? Andrei Alexandrovich Milaronov, Doctor of Biological Sciences, Professor of the Faculty of Bioengineering and Bioinformatics, Moscow State University, named after Lomonosov. Can you hear us? Thank you. Adam Michael Filippi, PhD, National Institute for Human Genome Research, United States of America. Can you see and hear us? And of course, the applicant herself is also present with us. To improve the quality of communication, I ask you, dear colleagues in the remote access mode, to turn off your microphones, but not to forget to turn them on when you are given the floor. I'll also inform you that audio and video recording of our meeting is being carried out, and live broadcast is organized on the St. Petersburg State University website. Simultaneous interpretation is provided. Currently, an email address is posted on the live broadcast of the meeting of the Dissertation Council, where during the meeting all the listeners can express their opinion and send questions to the applicant online. The questions should concern the dissertation and the current scientific discussion of the presentation. Technical service will forward the questions to me and I will voice them during our discussion. Questions might be related to the applicant's presentation and the content of the dissertation. And it is necessary to state the name, surname, title, and place of work of the author of the question. Questions not related to the scientific discussion, to the discussion of the dissertation, to the text and the assessment of the dissertation itself will not be voiced. In accordance with the procedure for awarding the academic degree of Candidate of Sciences, academic degree of Doctor of Sciences at St. Petersburg State University, approved by local regulations of St. Petersburg State University, hereinafter the procedure, the meeting of the Dissertation Council is considered competent if at least 2/3 of the approved participants take part in its work. Not less than 4 people. Our Dissertation Council consists of 5 people. All 5 members are present, including 3 members in the remote online mode. All members of the Dissertation Council are provided with audiovisual contact. Thus, there is a quorum. I establish the following rules of procedure for today's meeting of the Dissertation Council, with a total duration of approximately 2 hours. A brief report of the chairman on the documents submitted by the applicant for the scientific degree and their compliance with the established requirements. Answers to possible questions. Short presentation by the applicant for the scientific degree reflecting the main points of the research. Approximately 15 minutes. Questions to the applicants strictly according to the report, no more than 2 minutes per question. Answers of the applicant to the questions, no more than 5 minutes for all the questions. Speeches of all the members of the Dissertation Council, in turn, with their assessment of the dissertation research, and the applicant's presentation, with the summary of their position, questions and suggestions for the dissertation. Approximately 10 minutes per speaker. Speech by the chairman with the assessment of the dissertation research, about 10 minutes. Answer of the applicant for the scientific degree to questions and comments of the members of the Dissertation Council, no more than 20 minutes. Open discussion. Speeches at the request of those present at the defense of the dissertation, with summary of their position, of their specific questions or proposals for the dissertation, strictly on the topic of the research. No more than 2 minutes per person. At the same time, we ask everyone to register in the registration form and introduce themselves before speaking. Presentation by the chairman of question addressed to the applicant, which came in order to discuss the presentation and scientific issues following the live broadcast of the meeting on St. Petersburg State University website. I pay your attention to the fact that written questions that need to be answered for more than 2 minutes will not be voiced. For the applicant has only 2 minutes per question. Presentation of the scientific curator, no more than 2 minutes. Discussion by the members of the Dissertation Council before the open roll call. The sound of the broadcast is muted. Open roll call. Counting of votes by the chairman of the Dissertation Council with entry the result into the minutes of the meeting. Making a decision on awarding or not awarding an academic degree. Concluding remarks of the applicant, no more than 2 minutes. Dear colleagues, are there any questions or objections to the procedure? If there are no objections, I will proceed to the implementation of the rules of the meeting. Please turn off your mobile phones. And the colleagues who are in remote mode, I ask not to turn off the phones, but only to turn off the sound, in order to have the opportunity for prompt communication in case of technical failures. Thank you for your understanding. So I'm starting to conduct the meeting. Alla Alexandrovna Mikheenko, thesis for the degree of candidate of physical and mathematical sciences in the scientific major 030109 mathematical biology bioinformatics. On the topic, development of computational methods for the analysis and visualization of eukaryotic genomic assemblies. was accepted for defense by the order of the Scientific Secretary of St. Petersburg State University, dated 28th of October 2020, number 9608/1. Alla Alexandrovna Mikheenko wrote her dissertation research on the basis of St. Petersburg State University. The scientific supervisor is Pavel Persner, candidate of Physical and Mathematical Sciences, professor at the University of California in San Diego, USA. Professor and chief consultant of the Laboratory Center for Algorithmic Biotechnology of St. Petersburg State University. The number of publications of the applicant, in which the main scientific results of the thesis are stated, according to the presented list, are 4. These include the publication in peer-reviewed scientific publications from the list approved by the Ministry of Education and Science of the Russian Federation. In publication indexed in the scientific databases Web of Science and Scopus, all 4 publications. The applicant submitted to the Academic Secretary of St. Petersburg State University a complete package of documents for acceptance for consideration and defense of the thesis. All the documents correspond to Clause 12 of Section 3 of the procedure. All the documents submitted by the applicant, according to the information that I have received from the curator, meet the requirements and are stored in the applicant's certification file. Copies of them are held by the employee of the Department for the Provision of the Dissertation Council, who is now attending the meeting. Before giving the floor to the applicant, I would like to ask you, dear members of the Dissertation Council, if you have any general questions to the applicant, including whether there is a need to announce and consider the entire list of documents submitted by the applicant. I understand that there are no questions, so there is no need for it. Which facilitates the procedure greatly. No questions? Then the floor is given to the applicant. You have the floor. The time limit is 50 minutes. Good afternoon, dear colleagues. Today I'm going to give a presentation about the main provisions of my thesis. My today's presentation will be divided into 3 main parts, according to the 3 main tasks to which my work was dedicated. This is the analysis of the quality of eukaryotic genomic assemblies, the analysis of ultra-long tandem repeat assemblies, and the creation of new approaches to visualizing genomic data. So assessing the quality of genomic assemblies is one of the essential steps in almost any genomic project. There are 2 main approaches to that, depending on the use of reference genome. Reference genome is a high-quality assembly of the genome of an organism and a study or a closely related organism with which assemblies can be compared to obtain a more accurate pictures of their quality. The first group of approaches does not use a reference genome and includes basic statistics, analysis of assemblies based on the set of reads that we use to obtain assembly data, as well as the assessment of the assembly quality of individual genes, which can be of critical importance for further stages of the studies. The second group of approaches is based on comparing assemblies with a reference genome. The 3 main evaluation criteria are completeness of assembly, continuity, and correctness. The completeness of the assembly shows what part of genome was assembled, a continuity, how many fragments the assembly contains and how long they are, and correctness, how many errors the assembly contains in comparison with a reference genome. One of the standards of assessing the quality of genomic assemblies is the QUEST program created in our laboratory in 2012. It is important to note that QUEST was created primarily for the analysis of bacterial assemblies. In subsequent years, the development of sequencing technologies and the accompanying development of assembly algorithms led to the emergence of a huge number of assemblies of complex eukaryotic genomes, for the analysis of which there were serious limitations. Therefore, we decided to develop a fundamentally new version of the Quest program, Quest-LG, in which the problem that hindered the effective work with the eukaryotic genomes would be solved. First, the size of the eukaryotic genome is much larger than the size of the bacterial genome. Thus, new highly productive algorithm was necessary. Therefore, we completely revised the most time-consuming part of the algorithm alignment of assemblies to the reference genome and developed several heuristic procedures that significantly speed up the program. Secondly, eukaryotic genomes contain many repeated regions. Therefore, alignments of assemblies with the reference genome gives usually ambiguous results. In this case, number of different algorithms helped us to arrive at different alignment options for each contact per reference, and it can reach several thousand. It works rather quickly, even for long assemblies. The algorithm maximizes the percentage of reference coverage while minimizing the assembly errors. Also, transportable genetic elements have a huge contribution to the difference between the reference and the genome of the studied organism. We developed a procedure to distinguish true assembly errors from inconsistencies generated by transportable elements, and have added analysis of assemblies based off unique K-mers. Since transportable genetic elements are usually present in genome in many copies, although not each copy contains very few unique K-mers or K-measures, and they will not significantly affect the result of the metric. It is worth noting that a reference genome is often the combination of different sequencing technologies using manual correction of assembly errors, and the quality of the reference genome is unattainable in most simple genomic projects. That's why we developed the more realistic concept of assembly, which is complete and correct as possible for a particular set of reads and reference genome. We have taken into account the low coverage regions of the genome as well as long genomic repeats, a serious problem for the assembly algorithms, and often cannot be completely reconstructed. This slide represents the results of the Quest-LG program for 3 assemblies of the human genome obtained on the basis of reading of Oxford nanopore, and also for theoretically optimal assembly of the reference genome. As you can see, the indicators of completeness and continuity for theoretically open assembly and much closer to those of real assemblies than the reference genome. In the reference human genome, each chromosome is assembled into one contact, and this quality cannot be achieved using only one library of long reads. The slide shows the correctness metrics for the same assemblies. Here you can see that assembly errors has 20 times higher than in other assemblies. The correctness indices based on K-mers do not differ significantly. This can be explained quite simply. In here we can see the alignment of assemblance of reference genome. Erroneous contacts are highlighted in red. It can be seen that the majority of the errors are concentrated in the pericentromeric regions, which were not collected in the other assemblies. As mentioned before, errors in duplicate regions did not significantly affect metrics calculated from unique K-mers. Thus, the results did not differ that much. The centromeric and pericentromeric regions of the genomes have always been considered one of the most difficult regions to assemble. The second part of the report will be devoted to such sequences. The analysis of the assemblies of ultra-long tandem repeats. Tandem repeats are nearly identical sequential copies of the same DNA sequence. and the work is dedicated on the analysis of ultra-long tandem repeats, that is, repeats of hundreds of thousands of millions of nucleotides. Some of the longest tandem repeats in the human genome are in the centromeric and pericentromeric regions. Until recently, centromeric regions in the human genome remained unassembled. Only in 2019 did the centromere assemblies of the chromosomes Y and X appear in the fall of 2020, and the first release of a new reference human genome from the Telomere to Telomere consortium was released. And this success was made possible by the development of third-generation sequencing technology and the development of assembly algorithms, in particular the emergence of central flight programs and Mikheenko software for genome assembly from PacBio Hi-Fi, which provide high accuracy reads that combine a read length and a very low error rate, 1 error per 1,000 nucleotides. At the same time, the assessment of such assemblies is extremely difficult. Reference-based approach cannot be used in this case. Firstly, reference sequences are usually absent. Secondly, it was shown that the level of variability of the regions is much higher than the variability of the rest of the genome. For example, the length and structure of centromeric regions can vary significantly between two different people. The analysis based on the reads does not usually give the objective picture, for the alignment programs do not work optimally in the high repeat regions. The analysis of a separate gene assembly is also non-applicable. Thus, we decided to create the Tandem Tool software package, which combines the Tandem Mapper program for aligning readings on the assembly and the Tandem Quest program for assessing the quality of assembly and search for errors. As already mentioned, both standard programs for aligning reads and modifications designed to work with repeat regions may produce incorrect or erroneous alignments, especially if the assembly contains errors. Imagine an error occurred while assembling a long tandem replay or read, and the assembler miscalculated the number of copies, and several copies were missed or during the assembly. And since the repeat is highly repeated, different sections of the assembly have a similar match. In different error occurs, the position of the chimers in reads and assembly does not match, no alignment occurs, and no matter how similar the nucleotide sequence are in general. This approach allows a very low number of misalignments to be achieved. So if chimers do not have the same position in the assembly and no alignment occurs, it helps to decrease the number of misalignment. Here we can see the results of Tandem Mapper and other program on simulated data. We can see that Tandem Mapper did not make any errors in aligning reads to the section with assembly error, unlike other programs. And this is of special importance because incorrect alignment of reads leads that on further analysis it is very difficult to establish the assembly errors. This is why it's important to minimize the number of incorrect alignments. Tandem Mapper results are used by Tandem Quest to evaluate assembly quality and search for errors. Tandem Quest has two main modules. The first is used for calculating basic metrics such as coverage metrics, assembly breakpoint metrics, measure-based metrics, and pairwise comparison of assemblies. The second is designed to assess assemblies of centrometric regions and the metrics that are developed taking into account their structure and organization. In order to solve the problem and finding the error, we need to develop an algorithm that searches for breakpoints. The algorithm is used on the following observation, or the algorithm can't extend the alignment through the breakpoint of the assembly because of the difference in the distance between the k-measures and reading and the assemblies. Thus, if many alignments begin or end in a particular area, it can be assumed that the area contains an assembly breakpoint. For each of the k-measures, we calculate the alignment percentage which ends or begins in it. Consider this indication appropriate for a perfect situation where the read has been completely checked. If there are no errors, the measures will be similar. But if there is a breakpoint in the assembly, the percentage of k-mirror or k-measures, the alignment percentage will be much higher than in the perfect situation. In here we can see the example of assembly of the 8 chromosome. The blue graph shows ideal situation and the second graph shows the real situation. Very often the graphs correlate, but at a certain point we can see the rise of one of the graphs, which indicates the breakthrough. Thus, it shows that there is an assembly error. And detailed analysis showed that the assembler missed several copies of the repeat. Thus, in high repeat regions, it was possible to find the errors. The last part of the report is dedicated to visualization of genomic data. Visualization is an important part of any research as it allows you to discover new connection patterns and see a more complete picture. Many tools have been developed for visualizing genomic data, but they all have their different areas of applications and their certain limitations. Previously, there were no specialized tools for visualizing the assessment of the quality of assemblies based on the reference genome. We consider that the instrument developed by us will be useful for assemblers in order to find the assembly errors. In here we can see the example of how Eukaryotic program works to assemble the yeast genome. We see the alignments of the assemblies of the reference genome and the read coverage panel. The assemblers were not able to correctly assemble it. And in here we can see the sharp rise in coverage, which indicates the presence of high magnification repeat. More detailed study showed that there was a long tandem repeat at the position, which was not correctly resolved by genomic assemblers. We also developed the assembly graph browser for interactive visualization of a large assembly graph. The program presents a wide range of approaches to the presentation and simplification of graphs. The example can be seen in the slide. The assembly graph of the yeast genome with the edges of the graph corresponding to the nucleotide sequences and the vertices serve as connectors. The colored edges are repeated here. They have the red color and the unique alignments are black. Here is an example of visualization of the assembly graph of the human genome. The assembly graph contains several thousand vertices and is extremely inconvenient for perception. So we need to split the graph into several subgraphs and demonstrate the links between them. Since we know that our repeated edges usually correspond to sequences such as transportable genetic elements, tandem repeats, segment duplication, and if these elements are not of interest in a particular case, we can compress all repeat edges and get compressed assembly graph, which represent the structures of unique sequence. Also, AGB allows us to visualize the path in the graph for each contig, or attach the assembly graph to the reference using the QuostLG program. Thus, AGB extended the functionality of visualizing assembly graphs, allows us to work with large graphs containing thousands of vertices, and can be useful for both developers and users of various genomic assemblers. In conclusion, I would like to summarize. The program QuestLG was developed for assessing the quality of eukaryotic genomic assemblies, the creation of the Tandem tool software package, including the Tandem mapper program for attaching long error-prone reads to assemblies of Tandem repetitions, and the Tandem Quest program for analyzing and validating such assemblies, as well as the development of new methods of visualizing genomic data. Those include those such as the Eukaryotic browser for visualizing assemblies and AGB program for visualizing assembly graphs. I would like to thank all the colleagues from the journal Bioinformatics and my supervisor, Pavel Pazner. Thank you for your attention. Are there any questions to the speaker? I have a question. Can we have the slide with the 8th chromosome back, please? Yes, this one. Why? In the position with zeros, why is there a gap? It means that in this position, the alignment is not ended. Either there is no coverage there, or the application is not there, so it goes through the region. So we need to have a look at the graph coverage to understand what is happening in this section. If there are no more questions, then we move to the second part. The presentation of the speeches of the members of the Dissertation Council with their reviews of the thesis. We decided that the applicant will respond to the questions after each particular review. Since all the reviews were published on the St. Petersburg State University website. So I offer to state only the key points, questions or suggestions. Are there any objections? Well, sometimes I will violate this rule.
First defense, which is going on namely in Russian, not in English. So let's change the order a little bit, and Adam will be the first. Please.
Adam, the floor.
Yes, thank you. Can you hear me?
Yes.
Great. Well, I'll start with my summary of the dissertation and end with a few questions for Alla. First, I found the English portion of the defense very well written, with very few errors, so I appreciate that, Alla. Thank you for taking the time to make it easy for me to read. First, the relevance of this dissertation to my own work is extremely high. As Alla mentioned in her presentation, she has applied her methods to our efforts to finish the human genome, and as many of you know, the human genome has remained unfinished for over 20 years because of these satellite repeats around the centromeres. And Alla's work on Quest and Tandem tools and Tandem Quest was extremely critical for us finishing those regions of the genome. And as she showed some examples, for example, chromosome 8 there was other centromeres where her tools were able to flag errors that we were then able to fix. And it's rare to have graduate student dissertation work apply so directly to an important international project like the TDT project. So that's fantastic. The validation of the work I found was good, lots of simulation and validation. I have firsthand experience using these tools and I find them to work well. The novelty of the tools is interesting to me because there's very clever heuristics and methods applied in tandem tools combined with visualizations that make these errors very obvious to the eye. And there's a famous saying in information visualization that good visualizations make the answer obvious. And I think as you saw from that chromosome 8 example, the error becomes obvious when you plot these particular metrics that Alla has invented, and that's wonderful. So in conclusion, she contributed directly to the completion of the human genome with some of this work, and the standards of the dissertation are high by my evaluation. So my question for Alla is two parts. The first one is that QuAST-LG was designed for evaluation of assemblies, and we know the human genome has been incomplete. Many other genomes are incomplete, so we're often left with unfinished reference genomes. That makes more errors appear than are really there in the assemblies we're evaluating. And so we really have this need for assembly reference-free methods like tandem QuAST. So what's preventing you from applying tandem mapper and tandem QuAST to the whole genome for validation rather than focusing just on the satellite repeats?
На русском, да? Спасибо. В идеале, конечно, мы...
For your question, we would definitely like to combine these methods and use them for the analysis of the full genome, but now their productivity is not very high to work with the full genome. Since we concentrated mostly on centromeres alignments, and parts of the algorithm might be used for the analysis of the full genome, but certain changes are necessary which are not quite obvious. We might analyze the human genome, but it might take several days, and we cannot find certain errors which QuAST usually finds. So we cannot differentiate them. Inversions or other errors can be easier found in the course program, and maybe this part does not require any changes. But we can see an ideal possibility of using QuAST tools and automatic identification of the assembly regions and using the algorithm for the analysis of centromeres. Are you satisfied with the answer?
Yes, and my second half of the question is with regard to diploid genomes. So CHM13 is mostly a haploid genome. Chromosome X and Y are haploid chromosomes. How do your methods apply to diploid genomes, and will you have to make modifications in the future to account for validation of diploid genomes?
Да, у нас тоже есть такой ответ.
Yes, this is a very important issue nowadays, the work with the different diploid genomes, different haplotypes. And if we talk about the QuAST program, there are several options how we can use it. We can either work with the haplotypes already, or to compare within the reference genome, or with the use of visualization, we can see which parts are similar and which regions are different. We might use some haplotypes as reference and compare other haplotypes with them. Also, we can change the QuAST program using the assembly graphs and use the calculations in the graphs and apply them to the reference. Some metrics might change, thus the number which has to be close to 1 might increase depending on the difference of haplotypes, and the size of the assembly might change and the number of errors might increase, but some of them will not be errors but the real differences. Also, QuAST-LG has the program for the search of structural variations and deviations, which helps us to differentiate the assembly errors and the true variations between the organism and the reference. Also, we have certain algorithms which help us to differentiate between the errors and heterozygotic mutations. Since alignments are based on k-mers, we can always compare the distance between k-mers in the assembly and in the reads. And if there is an assembly error, all the k-mers, all the reads will differ by the same measure. But if we see that in half of the reads there is one option, or there is one distance between the k-mers, and in the second part of the reads there is a different measure, then we can see a heterozygotic option, which can be used with work or with haplotypes as well.
Thank you. That's the end of my questions.
That's it. Татьяна Валерьевна, будьте добры.
Dear colleagues, first of all, I would like to note that this thesis is a very interesting work dedicated to a very relevant topic. So all the issues linked to the assembly of repeated sequence is a very important issue, and many problems in genomic assemblies are related to the difficulty of these assemblies. Since my review is published and you had the ability to look through it, I'm not going to read it through. I would like to say that the main parts of the dissertation were very positively provided in the presentation of the speaker. And I would like to pay attention to certain questions that I had, but I would like to say that partly they have been answered. When describing the approaches of assessing the completeness of genome assembly, the author describes as one of the options the search for universal conservative genes, which are defined as a set of genes that are expected to in the same copy. So there is no duplication or no loss in a sequence genome for the corresponding phylogenetic group. And I would like to know what limitation of this approach the author sees from the biological point of view. Well, I suppose that we will do the question and then the answer. One of the most popular programs used for the search of such universal highly conservative genes is BUSCA program, which uses the genes that can be found in one copy only. These are 90% of the types of the specific phylogenetic group. It has certain limitation, of course. This set of genes is on one hand universal, but it is not equally well represented in any organism, and we cannot beforehand say how fully it is represented in an organism. Thus, a program underestimates the fullness of the genome, since not all regenes can be presented in the genomes, or it could have changed, or the prediction of the gene is not a 100% accurate procedure. And thus, non-model organisms showed that this completeness might reach about 50%, although the assembly is good. So we don't know which amount of number of the genes is lost during the evolution process and which amount is duplicated. So this shows either technical problems or the assembly of different haplotypes. So these are the major problems of such an approach. The next question is the following. On page 24, the author writes about the possibility of using crystal G for detection of transportable elements, and further indicates that an alternative approach would be the classification of these elements based on a genomic consistency, but this is beyond the scope of work. That I would like to hear which works outside the scope of study are meant here. The classification of transportable genomic elements is a separate task, and the golden standard in the area is the master, which provides full classification of all the genome elements. But first of all, it is rather slow, and then it searches for repeats, which it has in its database. So if some of the repeats are different only in this new organism, then the program will not find them. But there are some additional programs which allow you to create de novo repeat libraries and use them in combinations. The main fallback of the program, why we did not include it in our pipeline, was the fact that low speed, that is several days or even weeks for human genomes, and for model organisms there are ready-made results in the forms of programs which can be used and could help us to identify certain transportable elements. Also, there are programs for de novo identification of repeats and more specialized programs. For example, Utafarinder and others. But they're more specialized. This is why we did not include them as well. There is also the simplest way of finding repeats based on the coverage, but with the help of this approach, we cannot distinguish tandem repeats from transportable elements. So this approach is not universal, and we did not use it in the scope of our work. And my last question is mostly about the aesthetic concerns, mostly about the aesthetic part, about the visualization. Why does Icarus use blue for right contigs and orange for wrong contigs? Well, for the right, usually we use green. Well, we use green and red for right and wrong contigs, and blue and green for right and wrong contigs, which are which resemble each other. So if we can see that in some of the repeats it was assembled in a similar way, so we can visually emphasize it to show that the specific region was assembled in the same way. So this is why we use not a shade of green but blue to show which part of genome is similar and which part of the genome is different. Thank you. Do I need to read the conclusion, these words that we did not violate the procedure and that the work meets the requirements? Then I proceed. The presented thesis was carried out at a high scientific and methodological level and corresponds to world standards in the field of bioinformatics. Work is essential to the fundamental and applied science. Its results are highly appreciated by the scientific community. The validity of scientific provisions and recommendations formulated in the thesis is beyond doubt. The results of the work are presented in the form of reports at international conferences and scientific articles and prestigious journals, and is said by Web of Science of Scopus, cited hundreds of times. And current versions of tools are available at addresses that can be found in the review. The thesis of Alla Mikheenko responds to the key aspects of the procedure. According to the order 6291/1, according to the awarding of degrees of St. Petersburg State University, and merits the awarding of the degree of Physical and Mathematical Sciences, Specialisation 03/01/09, Mathematical, Biology and Bioinformatics. I am sorry, I forgot to mention Are there any online questions? Currently, there are no online questions. We'll be waiting for the questions. If there will be any questions, I will announce that. And now we move on according to the protocol, and we give the floor to Andrei Mironov. Well, I would like to say that the work, as all the works of this laboratory, is outstanding, and I'm not going to say the same what the colleagues said about the positive side of the work, but there are certain questions and comments concerning the thesis. The first question is the following. The way to assess the quality of an assembly is to compare the assembly with a reference genome. However, what is the reference genome? It is the genome which was assembled by somebody. Therefore, the assembler shows what was assembled by a different assembler. So the naive reader might assume that assessing the assembly quality by comparison with the reference assembly is a comparison of one assembler with the same standard version. Thank you very much for your question. Reference genome is a separate notion, and it has to be obtained as a result of a certain assembly. But the principal difference, which maybe was not fully indicated in the thesis, is that the reference genome has a higher quality. So we ideally want each chromosome of the organism be assembled in one contig, in one alignment. And in order to achieve such quality, we need to use different technologies and combinations, and to conduct some manual work sometimes on correcting the errors. And the reference genome, especially of model organism, is currently improving, becoming better, and certain patch comes out which cover the gaps in the sequences and or which correct the errors. But yes, of course, reference genome is also the result of genome assembly, and it might be assessed and validated with other approaches which are not based on reference genome. So there has to be the quality check for reference genome as well to find errors in it as well. Thank you. The second question was already mentioned, but my context will be a little bit different. Genomes, eukaryotic genomes, are usually diploid. Sometimes the maternal set of chromosomes may differ from the paternal one by a noticeable number of rearrangement. It has maternal and paternal chromosomes. And cancer genomes also have significant number of rearrangement. And the majority of the human genome were before used as cancer genomes. But now it is better. It has a lot of rearrangements, these cancer genome. But at the same time, the reference genome is usually haploid. Although recent assemblies are not continuous and they allow for variation, the use of graphical representation of genomes, which might show not only a haploid, diploid, but also states, different individual variations. How can the proposed methods be used for a graphic representation of genomes, for example? As far as I have mentioned, we can change the program in order to work with the assemblies. which are shown as graphs. But in order to perceive the graphs of the reference genome and take into account the two options of a possible construction of a certain sequence, more changes in the algorithm are required. Theoretically, it is possible, and maybe new tools might be required aimed at the alignment of graphs. So these are possible changes. And taking into account these individual variation of haplotypes is very important. And there are certain ways of changing our algorithms to use them with the graph perception. Further on, I have certain reference in use to the editing. Well, not always. You show, if we talk about literature reference, sometimes you do not indicate the year of publication, and there is no standard of giving reference. Sometimes the figure captions are not standardized. I suppose that I have voiced the major comments, and I'm satisfied with the answers. So I would like to say that it is great that the applicant is published, well, the results of the work are published in good magazines, journals, scientific journals, and the work corresponds to the procedure. Mikheenko's dissertation on the topic development of computational methods of the analysis and specialization of eukaryotic genomes meets the basic requirements established by the order number 6821/1 dated the 1st of September 2016, the procedure of awarding academic degrees at Specialisation State University. And applicant Alla Mikheenko deserves to be awarded the academic degree of candidate of physical and mathematical sciences in the major of 30109 mathematical biology and bioinformatics. Clause 1 of the procedure was not violated by the candidate for the degree.
Спасибо большое. Mikhail Sergeyevich.
Thank you very much. And now we give the floor to Mikhail Gelfand. Good evening, dear colleagues. My part is rather easy because I suppose that everything has already been mentioned by my colleagues, I suppose. Following the discussion that we had concerning the previous comments about the color graph, whether it was checked on the people who do not see the colors clearly, because if something is identified with green color and with blue color, certain people simply do not see the difference between these colors. We once had such a situation when we were doing a color scheme. One of the managers did not see the difference because he simply could not identify these different colors. So was it checked on the website that can fixate different types of people viewing different colors? No, we didn't check it. We used the simplistic approach, saying that green is correct and red is incorrect. And possibly There are standardized websites which allow you to see how these pictures are perceived by people with a certain disability of color perception. And the percentage population is rather high. And we have a colleague in the laboratory who had such problems with perceiving these colors, and it was only by accident that we found that. Well, that was one of the least important comments. On the whole, as the reviewer of the dissertation that is based on the published articles, it's a bit difficult for the reviewer because the reviewers of the journals, the editors of the journals where the articles were published could see all the major flaws if any were present. So I see no major flaws in the thesis. And all my comments are related to the idea of editorial part. Well, I paid attention to the literary reference list, including the publications of the author. They all have different... they are all presented in different styles. And it is very interesting how you were capable of using so many fonts in this work. But this is a pure editorial matter. Well, as a rule, it is typical for the dissertation based on articles. There is no good introduction, so there is no good proof or basis that would provide the background for the project that the author provides. So there is no internal unity shown, although we understand that all these components form the part of one work. This is related to many projects because the applicants, when making a thesis, the applicants do not usually show how all the elements of a larger project substitute each other and might be used in tandem. The overview is rather interesting, because you have some information about the basis molecular biology. So why should the reader understand how is the operant structure in bacteria presented? Because we don't need this information in the future. The same problem arises with the glossary. The glossary itself is very useful, but sometimes there are some generally known terms such as mutation, definition, but although there are some terms like the word unit is not defined in the glossary. So the glossary is done for its own sake, maybe, so there is no point in having it in the thesis. As for the major issues, we discussed some of my comments, some of the comments previously. As for repeats in misassemblies, they cannot be found because this limitation is imposed by the authors themselves. Well, I disagree. Well, I suppose that I disagree with the applicant in the idea that Riv-Master doesn't work in this way. But if we consider the coverage, if misassemble is in the region of high coverage, so these regions could be compared to each other and thus to see the repeat. Well, this is a separate project maybe, but certain simple things of such a way could be used that would result in the improvement of the system. So I think that this list limitation is a bit artificial. And even the simple filter on the repeats could be very useful. That was previously mentioned, so I do not require your answering my comment. I also mentioned some of the editorial things concerning the text itself. Well, I'm not going to repeat those. They can all be found in the paper back option of my review or on the website. But the contents of the thesis is great, and I would like to agree with Professor Mironov saying that the overall level of the dissertation is extremely high. And the dissertation in the line of all the other dissertation stands out. And the practical applicability was proved by the empirical measures used, the information used in the international consortium, in the international project. So it was already improved by the international scientific community. And the publications, the articles are of high standards, and the author of the thesis is the only author of the articles. So the name of the author is stated as first in the articles, and this is important for projects because many people participate in creation of the project. It is of importance to note that the author took part in other projects not linked to the topic of this work, which shows how broad is the sphere of interest of the author. And this does not characterize the thesis, but it characterizes the author of the thesis. So now I read the magic words. Let me find them. The thesis by Alla Mikheenko, Development of Computational Methods for Analysis and Specialization of Eukaryotic Genomic Assemblies, meet the basic requirements established by the order number 6A21/1, dated the 1st of September 2016, on procedure for awarding academic degrees in Dissertation University. Clause 11, the procedure was not violated by the candidate for the degree. I'm, by the way, very interested how many of the applicants are interested in actually knowing what is it, this clause 11 of the procedure, but whatever. But is it still not violated? And Alexandrovna Mikheenko deserves to be awarded the degree of candidate of physical and mathematical sciences in the speciality of 30109, mathematical biology, bioinformatics. Thank you very much. And as for the protocol phrase, well, I said the protocol phrase, didn't I? Well, I said all the magic words in the correct order. No Satan is to appear. So there are no questions online? Mikhail Sergeyevich gave a speech, so to say. I would like to say that I like this dissertation, this thesis pretty much, and the topic is very close to me. But I will also pay attention to the comments and questions, and so first of all, I'd like to pay attention to the presentation, first of all. Bioinformatics regards the measure of the human genome in certain millions, billions, and thousands, and 461 nucleotide. Well, this is impossible because the thesis states that the tandem repeats might be different with individuals, but this measure of human genome goes from one work into another because it gives such accuracy, which is impossible. One of the web slides gave the classification of different satellites. Micro, mini, and the classical satellite DNA. And the bioinformatics have a very formal approach to that, only considering the measure of the nucleotide pairs. Tandem repeats of humans of more than 100 nucleotide pairs. But if we talk about mice, there is a famous pericentrometric repeat, satellite DNA, all the parameters the same, but it has 21 nucleotide pairs. Formally, it corresponds to mini-satellites, but this is not a mini-satellite. This is just a remark, considering the presentation. As a form of the discussion, I would like to ask several questions. From the text of the dissertation, we might see that the author doesn't make the difference between the centromeric and pericentromeric tandem repeats. The normal tandem repeat is taken for alpha satellite of the human, and biologists know that alpha satellite can be divided into alpha satellite Y1, that's pericentromeric human repeat, and alpha satellite 2, pericentromeric, which does not have 3 box, and they have principal difference. If we take into account this parameter of protein. And referring to alpha satellite as the reference satellite, the minor satellite of the mouse has a monomer of 120 nucleotide pairs and not 170 nucleotide pairs as alpha satellite. As I have said, that in monomers in the pericentromeric repeats, more than 100 nucleotides is not a parameter. For assembly algorithms, maybe these differences are not important, but these are very important for the function of the centromeres and pericentromeres. And I hope that the future work will be aimed at creation of programs which differentiate different tandem repeats based on formal principles. This is just a comment, and I don't require an answer on that, but I'm very happy that somebody started research in this area. The program string decomposer requires monomers. For centromers, we know monomers. If we use alpha satellite as the standard repeat, does it mean that for other genomes, when we don't know the centromeric repeat, can the user, before using Spring Decomposer, should conduct the classification of tandem repeats in the first readers or contigs of TRF2, which doesn't take into account micro and mini satellites? Yes, Decomposer requires an input of monomeric sequence. And if we know the sequence for humans, then the task of classification of monomeric sequence is a specific task, large task. My colleagues at laboratory are working on improving String Decomposer program in order to solve the problem of classification of monomers, so that the alpha satellite could be used for input, a basic element, and all the possible repeats extracted from it, and the monomers could be classified into monomeric alphabet, similarly as was done for the human. But it is a rather complicated task, since monomeric sequences are very similar, and it's rather difficult to classify them. And this is a task related to the evolution of certain regions. So there are no solutions to the problem as yet, but we hope to obtain some. In the thesis, the unassembled part of the genome of human is about 10 or 15% of the genome. But I would like to say that the terms are not correct a little bit. Usually, more significant figures are cited. So why this particular figure was used? This particular figure was used, 10-15% of genome was not assembled for the first version of the human genome, which was published 20 years ago. But on the whole, it's very difficult to assess the unassembled part of the genome. The part which has the repeat regions is much larger, but it includes not only tandem repeats, but many other repeats. But we wanted to focus specifically on the centromeric regions, and according to the latest assembly of the genome, human genome, the centromeric parts are about 6% of the genome. On page 41, the author says that the minimizer are more likely to be unevenly distributed on the assembly because of a local enlargement of the tandem repeats. Could you please specify why they will not be evenly distributed? Minimizers are chimers, which are found in a specific gap, and their distribution in the assembly has to be similar. In here we can see the density of minimizer on X chromosome, and we can see the decrease of about up to 0 in the centromere region, or because there are many similar chimeras which are filtered. 0.2% of the most often found chimeras. So in less repeat regions, the distribution will be different. The thesis by Mikheenko Alexandra on the topic Development, Computational Methods for the Analysis of Validation and Eukaryotic Genomic Assemblies complies with the basic requirements established by the order dated 1st of September 2016, 6821/1 on procedure for awarding academic degrees in Universitetskaya University. The applicant, Alla Mikheenko, deserves awarding the academic degree of candidate of physical and mathematical sciences in the major 030109 mathematical biology bioinformatics. Clause 11 of the procedure is not violated. Now we move on to the questions that we received online during the live broadcast. So I need to ask the members of the council if they are all satisfied with the answers of Alla Alexandrovna. Nod your head, please. Thank you. No online questions? There are no online questions. No questions. I thought you said that there are no questions. Great. We're so happy that there are no questions. Now, those who are present but do not form part of the Dissertation Council might be given the floor. It seems that there are no questions and comments, so we move on. Since we conduct our meeting with the participants who are in remote access mode, so I need to ask the members of the Dissertation Council, those who are present and those who are in remote mode, any questions unanswered, which are unanswered, or some unclear topics related to the thesis. Well, I'm absolutely sure that there are no questions. So we move on to the next. The review of Pevsner Pavel. The dissertation of Mikheenko is dedicated to the effective computational methods for analysis and visualization of genome assemblies. Genome assembly from different reads in one of the most important tasks of bioinformatics. At the same time, the assessment of the quality of genome assembly is an important part of any genome project. The new technology of genome sequencing and algorithm requires the development of new methods, analysis and validation of the new assemblies, which allow to validate the results obtained, find errors, and find the optimal assembly strategy. The work of Mikheenko is focused on the development of algorithms, can quickly, effectively work on long eukaryotic genome assemblies. The main results of the work of Alla Mikheenko are QuestLG, Icarus, and Tandem tools. This software is widely used by the researchers all over the world in different genomic projects. The cycle of works, including the articles on the development of QuestLG and Icarus programs, Alla Mikheenko was awarded with the award of St. Petersburg State University 2018 for the influence on the science of young researchers. During the work on the thesis, Mikheenko demonstrated the ability to analyze great amounts of theoretic and practical material well, provided the information on a practical basis. The methods used demonstrate wide knowledge in the computational biology. The scientific results presented in the thesis were published in 4 articles indexed in the Web of Science and Scopus and have a high citation rate. The obtained results were cited on international conferences on bioinformatics. The topic of the thesis is relevant, and the obtained scientific results are of practical importance and scientific value. The dissertation of Alla Mikheenko presented complies with the procedure, and it can be awarded the right for defense. The scientific supervisor, Professor Pavel Arkadjevich, the candidate of mathematical scientist, the main candidate of the Laboratory of Technologies in Petersburg State University, professor of California University, San Diego, USA, the head of the National Center of Mass Spectrometry, USA. And here we're offered where do we have the time for discussion, I wonder? Well, my paper says that we need to move into roll call, but I think that we need some time for discussion, don't we? Well, if I missed the moment where we had to, when Mikhail Sergeyevich spoke, I would like to say a few words about tandem repeats myself. So, the floor is mine now. I will say the key points in English.
Tandem repeats all my life, and I do believe that, you see, molecular biology and bioinformatics and all the modern biology, it is focused on genes. But genes are approximately the same and very conservative. What is different? But zoologists, they recognize organism because of shape. And all the modern biology say nothing about shape, how shape is going on. Leg and hand has the same differentiations, but different shapes. And I do believe that this code is in the tandem repeats.
But unfortunately, it is impossible to prove that because the sphere of tandem repeat is a dark matter of genome. And why ask this question to Alla? What we do, we try to classify the tandem repeats, not to assemble them of different organisms. As for the human, who is the modal object of this work, according to our preliminary data, the stage of the difficulty of the tandem repeats of a human is much higher than the difficulty level of the same tandem repeat of mouse, for example. And the C paradox, which is well known, is also the repeat assembly, mostly pericentromeric repeats. And it's a pity that throughout my life I didn't have the ability, and maybe I will not have the ability, that the form of the organism can be found in the tandem repeats. Because about 6 months ago we had the article that all tadalado and euchromatines are small logarithmic differences, but the larger 3D structure of chromosome is formed according to the tandem repeats. But throughout my life, I couldn't get to these tandem repeats because Hi-C doesn't see them, so it is not present in genome. So I'm extremely happy to see this thesis and this work today, and I'm happy that bioinformatics were able to see those tandem repeats. It's a great pity that so far only formal limited characteristics were provided. I fully understand that in order to create these algorithms you need to limit the field and to make the field specific. But I hope that sooner or later humanity will prove that the shape of organism can be found in tandem repeats. Thank you very much. So now I return to my image as the chairperson. So we did not require the break to discuss the result of the work. Ask the members of the council, maybe they do require the break. So first the dissertation council follows the procedure and then we will move on. Do we have to turn off the broadcast and discuss? No, not necessarily. We can just vote. So, open roll call. We vote on the result of the defense and make a decision on awarding or not awarding the academic degree to Alla Mikheenko. Open vote or roll call. According to the decision of the Dissertation Council on awarding or not awarding the scientific degree, is considered positive if more than half of the members, but no less than 3 members of the Dissertation Council voted for it, who were present during the meeting according to the clause 23 of the procedure. Member of the Council, Matveyeva Tatiana Valeryevna. Member Matveyeva Tatiana Valeryevna, 4. Member of the Dissertation Council, Mikhail Gelfand. Turn on your sound, please. Well, he said votes for. Andrei Mironov, 4. Adam Philippi?
Yes, I vote for awarding the degree.
And me, the chairperson, Podgornaya Olga, vote for. Thus, esteemed colleagues and guests, I would like to announce that from the 5 members participating in the vote, 5 people for, against none, abstained none. The decision on awarding to Mikheenko Alla Alexandrovna the degree of candidate of Physical Mathematical Sciences in Scientific Speciality, a major of 30109 Mathematical Biology Bioinformatics, is adopted. Dear colleagues, due to the fact that our meeting is held with the participants in the remote access mode, do the members of the Dissertation Council or the applicant or other people present any questions, comments on the procedure of the meeting? And so no comments and objections. And the floor is given to the applicant for the final word. I would like to thank once again all the participants, the members of the Dissertation Council and the chairperson for your help and for your work. I would like to thank my colleagues from the Center of Algorithm and Biotechnology and my supervisor, Pavel Persner and Alla Lapidus, and I'd also like to thank my family and my husband for support. Thank you all. Thank you, dear friends. Now you can turn off the sound and the video. The meeting is closed.